of the Department of Psychiatry at UC San Diego and is the inaugural holder of the Igor and Joanne Grant Endowed Chair in Psychiatry. His research is focused on developing novel treatments and understanding the neurophysiology of these novel interventions for severe psychiatric disorders. He holds or has held numerous federal operating grants and awards, including numerous grants from the NIH and CIHR. He has received over 90 million in research funding and has over 500 peer-reviewed publications. He's an editorial board member for Biological Psychiatry, and he is an associate editor for Clinical Neurophysiology. He is also a member of the Brain and Behavior Research Foundation, formerly NARSAD Scientific Council, and is a Clarivate highly cited researcher. The floor is yours, sir.
Thanks very much for the introduction and for the invitation to present. These are my disclosures. As mentioned, I have received funding through the NIH, through CIHR, which is the Canadian equivalent of the NIH. I spent the first 50 years of my life in Canada, and was very grateful to receive funding through CIHR. Wellcome Foundation is an international foundation in sports science in Canada. The Temerty family, incredibly generous family, one of the most philanthropic families that I've ever had a chance to work with. Amazing people. They also gave a substantial gift and have the Temerty School of Medicine named after them. The Grant Family Foundation squared, because I've received two philanthropic gifts from two different Grant families. Igor Grant, who is the former chair here at UCSD of the Department of Psychiatry, and the Grant family, Doug Grant, who is the patriarch and a very generous philanthropic family in Canada, Camp, who gave generously to help us build a neuromodulation center. I've also, here in San Diego, I should say, I've also worked closely with MagVenture and Brainsway. MagVenture for an NIH-sponsored trial that I'll talk about today, Brainsway for industry-sponsored trials that we've worked on in the past. I won't be talking about those today, and I've also served as a scientific advisor for Brainsway Inc. Okay, let's get started. So most of you have obviously are familiar with depression, but what you may not fully appreciate when it comes to depression is that depression is a very difficult to treat condition. In fact, if you really carefully look at the data, a significant proportion of the population who are put on antidepressants do not fully respond to antidepressants. And that number varies depending on what you define as being response. But there was a big trial that was published over 20 years ago now, the STAR*D trial. Cost the NIH over $50 million to run, and was, I think, pivotal in many ways, but one of the key ways is to demonstrate that there is this subpopulation of patients, upwards of over 35%, who do not fully respond to antidepressant medications. And what I mean by that is that they achieve sometimes partial response, sometimes no response at all. But if you actually look at the placebo data, and I'll get to those in a minute, you realize that level of response after 2 failed trials of antidepressant medications is no better than placebo. So what this emphasizes is that if you're diagnosed with depression, you have 2 trials of antidepressant medications, your chances of response are high. But if you haven't responded after 2 trials, the chances of response drop to something that is probably no better than chance, or probably no better than placebo, I should say. A lot lower than the chance. And oftentimes what happens with these patients is we put them on medication after medication after medication, as the last picture demonstrates. This is a patient who was on a panoply of medications to try to treat their depression, but they were still very, very persistently ill. Okay, so speaking of placebo, we published a trial years ago looking at placebo rates in treatment-resistant depression, which is defined as two trials. If you haven't responded to 2 trials of antidepressant medications. And what we demonstrated is that in a treatment-resistant population of patients, that is patients with severe symptoms, severe depressive symptoms, who had not responded to antidepressant medications, the chances of responding to placebo were about 1 in 7, so about 13%, which puts it right at around the range that we see in the STAR-D data, which is why I said it was really difficult to demonstrate that beyond 2 trials of antidepressant medications, your likelihood of improving compared to placebo is minimal. This is a paper that was published incidentally by my clinician scientist. We spent years on this because we had to collate all the data, really try to identify the question that we were trying to answer, that is placebo response to treatment-resistant depression. But it's a complicated subject, and pulling all this data together and publishing it was a feat of considerable effort, and that's what Brett, who's now a faculty member at the University of Toronto, did exceptionally well. This is another paper by another outstanding trainee of mine, Helena Kim. What you'll see in my subsequent slides is me highlighting the tremendous group of people that I had a chance to work with over the years, including faculty here at UCSD. I've been at UCSD 5 years. Helena was one of those trainees who, you asked her to look into something, and within a few weeks you had a paper in your hand. That's how productive she was, and continues to be. Now she's faculty as well at the University of Toronto, and she looked at the efficacy of antidepressants in 2 populations. Population 1 is population of patients who had comorbid medical conditions with depression, and a population that had comorbid psychiatric conditions with depression. What she found was that the response to antidepressants in these 2 comorbidities, again, depression with another co-occurring psychiatric disorder, and population of patients with a co-occurring medical disorder. And what she found was that the efficacy dropped off considerably. It was not very beneficial to be on antidepressant medications when you had co-occurring conditions. All this to say that it does speak to the mutedness of antidepressant medications in this patient population. Even further, if a patient population, this is the first of our faculty members who we managed to recruit from Canada, Corey Weissman, who was my trainee in Toronto. He's now head of our interventional psychiatry program here at UCSD. He's outstanding, and this is just one of the papers that he worked on for years. And this is a paper that is actually a follow-up to the STAR-D data. So I mentioned a big STAR-D data set, 50 million. It's been data archived. We asked for the data. When the study was conducted, it took 4 or 5 years, and this is in the late 90s, early 2000s. Our ability to track and collect data at that time was mostly through Excel, and so we asked from the data archive this Excel spreadsheet, and it took us a year to simply pore over it and get it in order. And then once we had it in order, we analyzed the data, and what we found was that antidepressant response also decreases significantly when people have co-occurring suicidality. That is, if you're depressed and you have suicidal ideation, the likelihood of you're going to respond to antidepressant medications actually drops off, and it drops off after the first antidepressant trial, meaning that the first antidepressant trial is effective. But unlike with STAR-D, where the second antidepressant trial was also effective, our data suggests that the efficacy of antidepressants are also significantly muted even after 1 trial antidepressant medications, meaning that suicidality in and of itself predicts poor treatment response. So what to do about that population, that very large portion of patients? I sometimes say that in San Diego, it could be as much as 80 to 100,000 people, an enormous number of people. What do we do with those patients? Well, this is one of our most efficacious treatments. It works about 75% of the time, but it's also one of the most controversial treatments, and that is ECT. ECT is a treatment that was developed in the 1930s. It's still a mainstay treatment for severe psychiatric illness, particularly for patients with suicidality, but it has its shortcomings, and those shortcomings are that it is significantly stigmatized. It's one of our most stigmatized or vilified treatments that we have in psychiatry, but it's also associated with memory side effects, and those 2 are related. That is, that memory side effects tend to prevent people from wanting to consider ECT, and it's only used in a small proportion of patients. Some data that I looked at suggests that only about 1% of treatment-resistant depression. So to use my San Diego data, probably only about 800 people out of those 80,000 would get ECT in any given year, leaving a substantial proportion of the population untreated. This is where transcranial magnetic stimulation comes in. This is what we're experts in. Transcranial magnetic stimulation is a magnetic field that was developed in the 1990s to treat depression, the early studies in the 1990s, and then there's been hundreds of studies since then showing considerable efficacy of transcranial magnetic stimulation for the treatment of depression. We stimulate the prefrontal cortex, and we stimulate the prefrontal cortex through different stimulation paradigms, but it only takes a few minutes a day. It's every day for about 4 to 6 weeks. And if you look at the efficacy data, you'll see that the efficacy and this is in treatment-resistant depression. So remember, these are patients who have not responded to antidepressant medications. It works about 50% of the time in that patient population. This is a large meta-analysis that we published back in 2014. Some additional trial work that we had conducted, paper by my very good friend and former fellow, Dr. Dan Bloomberger. Dan published this trial on 121 patients, and what he found was similar to the previous findings, that the results were muted, but they were enhanced, that is TMS, when we stimulated both sides of the brain. So here's an example of one side of the brain, but when you stimulate over both sides of the brain, you get a significantly enhanced response compared to placebo. TMS in and of itself is not only effective for treating depression, but when we reanalyze the data, again, this is Corey's work, when we reanalyze the data specifically in relation to suicidality, we also found that suicidality was significantly better when we're treating with so TMS is not only an effective treatment for depression and for depressive symptoms, but it also significantly improves the remission, or produces remission, of suicidality in patients with severe depression, specifically when it's delivered bilateral in that bilateral orientation that I mentioned earlier. There have been advances in transcranial magnetic stimulation. So the traditional transcranial magnetic stimulation that was approved by the FDA back in 2008 involved single pulse stimulation every 100 milliseconds to the dorsolateral prefrontal cortex. So 100 milliseconds is 10 hertz. We deliver 10 hertz to the dorsolateral prefrontal cortex for a period of up to 30 minutes. When that's delivered, we produce response in and around 30% of the time, and remission in around 20% of the time, meaning that 1 out of every 5 patients reaches remission, no depressive symptoms, and 1 out of every 3 patients is a lot better, more than 50% improvement. Still takes a long time. 30 minutes is not easy to sit in a chair and receive this treatment, and it doesn't really get to what the mechanisms are of therapeutic response. So in 2005, there was this very interesting work that actually originated from tissue studies demonstrating that a particular stimulation paradigm known as theta burst stimulation was used to enhance neuroplasticity. And this theme of neuroplasticity will keep coming up in my presentation, to enhance neuroplasticity in the cortex of healthy subjects. The theta burst stimulation is called theta because it's delivered at a burst, which is 50 hertz stimulation, real fast stimulation, 20 milliseconds between pulses. But it's a burst of pulses. It's only 3 pulses that are delivered at, excuse me, at 50 hertz. And that burst of 3 pulses is delivered on a 5 hertz rhythm. So it's a burst of 50 hertz, 3 50 hertz pulses delivered every 200 milliseconds. To make things even more confusing, when you deliver this burst of pulses, you can deliver it in one of 2 ways. You can deliver it as a intermittent form, where you're delivering stimulation for 2 seconds and then you're breaking for 8 seconds, and then delivering stimulation for 2 seconds and then breaking for 8 seconds. Or you can deliver it as a continuous burst, no break, just continuous bursts every 200 milliseconds. Because of the evidence in tissue samples, they took it to the human, into healthy human subjects, and they showed that intermittent theta burst stimulation, this paradigm where you're delivering stimuli, 3 bursts at 50 hertz on a 2 hertz, on a 5 hertz rhythm, actually produces neuroplasticity in the cortex. By contrast, the continuous form of theta burst stimulation produces an inhibitory neuroplasticity, an LTD-like plasticity. as opposed to an LTP-like plasticity, which is what intermittent theta burst stimulation does. And so this was an important finding. It was published in Neuron, one of our most seminal papers, because not only does it have neurophysiological implications, it also has treatment implications. Perhaps most importantly, though, and I want to emphasize this, is that a large number of pulses can be delivered to the cortex in a relatively short period of time, 3 minutes and 20 seconds. It's a very short period of time to be able to produce neuroplasticity in the cortex, something that was previously unavailable with the 10 hertz stimulation, which produced inconsistent effects on neuroplasticity. There was a paper that was then published in 2014 by Liedall. This was a paper in depression. What they did was they randomized people to 4 different groups, treated them for up to 4 weeks. One group got continuous theta burst stimulation over the right dorsolateral prefrontal cortex alone, 1,800 pulses total. Takes about 9 minutes. It takes about 3 minutes to deliver that. The second group was intermittent theta burst stimulation, 1,800 pulses. Again, takes about 9 minutes to deliver that. The third group is continuous theta burst stimulation plus intermittent theta burst stimulation. This is basically bilateral treatment where we're stimulating with intermittent theta burst stimulation over the left dorsolateral prefrontal cortex and continuous theta burst stimulation over the right dorsolateral prefrontal cortex. And then the final group was a sham group where people got sham stimulation, not real stimulation, but the pulses number was the same. And as you can see in this slide, the group that got this bilateral stimulation, much like what I showed you earlier, demonstrated the most significant therapeutic improvement. But the ITBS group at 1,800 pulses also did substantially better than the other two groups, implying that there's something about intermittent theta burst stimulation, and specifically when intermittent theta burst stimulation is combined with continuous theta burst stimulation over the right and left side of the cortex, where you get the most substantive therapeutic improvement in patients with depression. We picked up on those findings and decided in collaboration with Jonathan Downer, we being Dan Bloomberg and myself. Jonathan Downer is a friend and colleague. He was the head of interventional psychiatry at a neighboring hospital in Toronto, and we conducted one of the largest clinical trials in the field of psychiatry, but certainly the largest with TMS, and it was a non-inferiority clinical trial. For those of you who are statistically in the know, non-inferiority clinical trials are designed to compare the clinical two treatments and establish clinical equivalency between the two treatments. The purpose of this study was to really demonstrate that theta burst stimulation was as good as the standard FDA-approved treatment, which took 10 times longer. So intermittent theta burst stimulation takes 3 minutes to deliver, and the standard FDA high-frequency left stimulation, which is 10 hertz, takes about 30 minutes to deliver. And so what we found here was that the treatments were, for all intents and purposes, identical, which means that theta burst stimulation is equivalent in efficacy to high-frequency left stimulation. And it also means that you can deliver a substantive increase in the number of treatments over a course of the day. If the treatment only takes 3 minutes as opposed to 30 minutes, you can treat many more patients over the day. And this actual finding led the FDA to approve theta burst stimulation for the treatment of depression. And it's the reason why many patients across the country receive theta burst stimulation for depression, including patients here in San Diego in our clinics. So the trial itself, the 3D trial, was published in Lancet in 2018. Stimulation of the left dorsolateral prefrontal cortex. What we found was response rates of about 50%, remission rates of about 33% higher than the previous trials, and demonstrating that about a third of patients had no depressive symptoms and 50% of patients were significantly better. There was a follow-up study to this one published just a few years ago that looked at geriatric depression. Because geriatric depression tends to be more resistant, we decided to compare bilateral treatments in these patient populations, but using the same non-inferiority clinical trial. We found, again, equivalent results. That is that theta burst stimulation was really beneficial in these patients. Short treatment, about 10 minutes long over the left and right prefrontal cortex to achieve therapeutic efficacy in patients with depression who were over the age of 60. And that was called a 4D trial. There's been considerable innovation since those trials were published. The most notable of those is the Stanford neuromodulation therapy, which was led by Nolan Williams and his team up at Stanford. And it's called Stanford because it's from Stanford, a neuromodulation therapy, but it's an accelerated form of theta burst stimulation. And it was based on the fact that if you can deliver one treatment for 3 minutes, why not deliver multiple treatments a day and try to get people better faster? And so involves 10 treatments a day over the course of 5 days. And there's a spacing of about 50 minutes between treatments. And the reason for that spacing is because it's been demonstrated neurophysiologically that there is an accretive effect of repeated stimulation on neuroplasticity in the cortex. The other major advantage of this treatment is that it precisely targets the prefrontal cortex based on the circuitry between the prefrontal cortex and the subgenual cingulate and the subgenual cingulate cortex. That dorsolateral prefrontal cortex subgenual cingulate connectivity has been demonstrated to be associated with therapeutic improvement in depression, and being able to target that circuitry was seen as a significant value add. In that trial, there were several of them. One was a small open label trial. Another one was another open label trial that was published in 2020, and finally a 2021 paper. Relatively small studies, but still producing significant therapeutic improvement, demonstrating that this accelerated treatment, again, over the course of a week, as opposed to over the course of 4 weeks, resulted in significant therapeutic improvement in patients compared to sham. Okay, well, I've now summarized some of the treatment effects, but what do we know about the mechanism of action? And as I mentioned earlier, that's where we start to really narrow in on or zero in on this concept of neuroplasticity in the cortex, but there are also other mechanisms, including inhibitory neuroplasticity in the cortex. We had endeavored to evaluate a marker of inhibition in the cortex. And this is a neurophysiological marker inhibition. This is the work that I did with one of my former postdocs, Andrea Levinson, who's a psychiatrist at the University of Toronto. She's now in charge of the College Mental Health program at the University of Toronto. But in early days, in the early 2000s, we had worked together to evaluate the neurophysiological circuitry of major depressive disorder, and specifically looking at different populations of patients. Populations we looked at were resistant depression patients, were patients who were unmedicated but had a history of depression, patients who were medicated but responded to antidepressant medications, and healthy subjects. So 4 groups of patients. What we found was intriguing. We found that it was, in fact, the treatment-resistant depression patients who had the most substantive inhibitory deficits. So there's something about the phenotype of resistant depression, that is, patients who were not responsive to antidepressant medications, which pointed us in the direction that these folks have an abnormal neurophysiological circuitry that needs more advanced care than just simply antidepressant medications. But the question that remains, quite frankly, is that when we endeavored on these trials, we looked at the motor cortex specifically, but any psychiatrist that you ask, where do you think depression, if you will, exists in the brain, their response will more than 9 times out of 10 be it's in the prefrontal cortex. The prefrontal cortex is the last part of the cortex to evolve. It has the most complex circuitry in the brain, and it is associated with reasoning, with cognitive performance, with the ability to relate socially. So the prefrontal cortex is a major target for patients with depression, which is why we've been treating it with TMS, that region of the brain, applying TMS to that region of the brain. These studies were in the motor cortex. And so we really wanted to understand the physiology in the prefrontal cortex because that was the site that we were also stimulating. It is not trivial. When we looked at physiological circuitry in the motor cortex, we could simply record hand muscles. But in the prefrontal cortex, it requires us to record complex physiology that involves using TMS, not as a treatment, but as a physiological tool, and stimulating the dorsolateral prefrontal cortex. And the motor output is electrophysiology, it's EEG. Now, any engineer will tell you when you stimulate with a big, heavy magnetic field that can be upwards of 2 Tesla over a really small electrode that's recording really subtle circuits, you're going to run into the problem of artifact. And so we had to overcome that artifact. And fortunately, we had the chance to work with incredible engineering teams, including teams in Finland and in Australia. And we were able to overcome that. I'm summarizing and skipping over years worth of work, but I will tell you that we recorded these interesting neurophysiological volleys. And over time, it's been shown that those neurophysiological volleys, including the N45, the P60, and the N100, when stimulated over the dorsolateral prefrontal cortex, translate into different physiological signals that are related to neuroplasticity and neural inhibition. And so we looked carefully at these neurophysiological signals with Daphne Voinescos, a clinician scientist trainee of mine. Now she's associate professor at the University of Toronto. And Daphne and I looked at these different waveforms and found that these different waveforms not only were associated with the condition of depression, that is, that there were abnormalities in patients with depression, but after we treated that those changes were normalized, and normalization of those changes predicted therapeutic response, which was exciting to us, and it pointed in the direction of what are the targets of treatment response in depression. And this is just an example of that N100 response. A dampening of that N100 response resulted in a significant correlation and relationship to improvement in therapeutic symptoms. Okay, I talked a lot about TMS so far, but I haven't talked about magnetic seizure therapy. And magnetic seizure therapy is a variant of TMS, but it's also a variant of electroconvulsive therapy. It's a variant of TMS in the sense that we're using a magnetic field to stimulate the cortex to produce therapeutic improvement. It's a variant of electroconvulsive therapy because that magnetic field is actually used to produce a seizure. And the reason when I was first introduced to magnetic seizure therapy, which was right around the time that I was first introduced to TMS, there was this innovative group out of Columbia in New York that had reported the first demonstration of a seizure with a magnetic field, a very large TMS coil. Actually, it's almost like two TMS coils in one at super high intensity. And despite those high intensities, the brain is actually stimulated with actually a relatively low field compared to the amount of energy that's required to produce a seizure with electroconvulsive therapy. The benefit of using a much lower field to produce a seizure is that it actually limits the spread of of electrical activity throughout the brain, which actually tends to spare cognition, one of the major barriers to using ECT in patients with depression. And so this was work that was led by Holly Listenby's group. Holly is now the inaugural dean of medicine, I think at University of Phoenix, I believe. She just moved from the NIH about a year ago. And what Holly demonstrated was that with bilateral ECT, there was a significant spread of electrical activity throughout the brain, whereas with magnetic seizure therapy, that spread of activation was quite limited. Now, the timeline of magnetic seizure therapy has been complex, and I'll get to the timeline. But it was first developed in the late 90s, first patients treated in early 2000s. We see here that these treatments evolved, but has really been in the last decade that we've conducted these seminal trials, magnetic seizure therapy and depression. One of the open label trials that we conducted took us about 8 years to conduct with a team of investigators. And what we demonstrated was that magnetic seizure therapy produced significant rates of remission when the frequency was actually quite high, 100 hertz. And we produced remission rates that were, depending on how you looked at the data, sometimes upwards of 50%. Here is a battery of cognitive tests that we apply to these patients. There's a lot of data here, and you're not going to be able to necessarily rapidly interpret the results, but what it shows is almost neutrality on cognition. That is, that patients' cognitive outcomes were completely benign. Patients didn't feel like they were getting a convulsive therapy from a cognitive perspective. And if you actually do a deep dive, there were two indices that stand out. The first one is the autobiographical memory, which tends to deteriorate a little bit. And then there's the verbal memory, which tend to improve. The autobiographical memory test that we used back about a decade ago was actually shown to not be a good marker of autobiographical memory because that test tends to deteriorate with time. And so we have now advanced to a new autobiographical memory called the autobiographical memory test, which is a much more precise way of evaluating autobiographical memory, which is not time sensitive. And the verbal memory test actually improved, as I said. So complete neutrality in terms of its cognitive outcomes. It also had effect on suicidal ideation, but specifically, we saw that lower frequencies had more beneficial effects than higher frequencies, although we may have been fooled by the relatively small samples in these groups. We'll get to that more in a minute. Paper that was published in 2024 by Holly's group, relatively small sample by randomized control trial standards, about 80 subjects, showed a very similar efficacy to electroconvulsive therapy, again with almost neutral effects on cognition. But this was a small study, and the pivotal trial I'll get to in a moment. Before I do, this is a paper that was published. One of our research scientists here at UCSD, who was formerly my PhD student at the University of Toronto, he's a biomedical engineer, Yiming Sun, and Yiming looked at the neurophysiology. I was mentioning my background, my PhD was in neurophysiology, and neurophysiology has been woven through all of our trials to be able to predict therapeutic response. And what we find here is that the extent of inhibitory neuroplasticity in the cortex actually predicts MST response, and specifically predicts it with a real high degree of accuracy, almost 90% accuracy. So the better your inhibitory neuroplasticity, the more likely MST produced suicidal ideation or remission in patients with depression, which we found is a pivotal finding. That's the reason we were accepted into such an impact journal. Yiming also demonstrated that magnetic seizure therapy induced neuroplasticity in the cortex, excitatory neuroplasticity, as well as inhibitory neuroplasticity in the cortex. And that led to a very large trial, a 5-year funded NIH trial. It actually took us over 8 years to complete this trial. We just completed the trial. The trial has been submitted in the last few weeks. So in terms of timelines, and to give you a sense as to how persistent we were, we first treated our first MST patient with depression back in 2011. That trial wrapped up in 2018. In 2017, we used the data from that trial, as well as another trial that I mentioned earlier, the DENG trial, as confirmatory data to be able to apply to the NIH under these confirmatory mechanisms, notice of awards. And the NIH allowed us to apply. We were lucky enough to get funded on the first round. And it was a large trial looking at randomizing people to either electroconvulsive therapy or magnetic seizure therapy in a double-blind design, and it was designed as a non-inferiority clinical trial. We weren't looking to see if magnetic seizure therapy is better than ECT. We were just looking to see if it's equivalent in efficacy. That's part one. But this was a gated trial, so there was a part two. And the part two, as you can imagine, based on everything I've said, is superiority statistically in terms of cognitive output, cognitive effects. So it was a complex trial to design statistically because we wanted non-inferiority from clinical efficacy perspective, but superiority in terms of cognitive outcomes. As I mentioned, that trial took place between two sites, the Center for Addiction Mental Health in Toronto and UT Southwestern in Dallas. In 2020, I was the PI of this trial. I moved to UCSD. UCSD Department of Psychiatry was not equipped to handle a trial of this magnitude when I originally arrived. That trial was eventually included here at UCSD, and we contributed a small but important proportion of the patients in this trial, getting us up to near our target. The other advantage of having UCSD on board is because we got hit by COVID, as we all know, in 2020, which significantly slowed our ability to recruit. In fact, it paused the trial for over a year because of the risk of COVID in bag mask ventilation. And so they paused the trial, we got it restarted, and the addition of the UCSD site helped accelerate recruitment to get to our final endpoint. A total of 239 patients were allocated to the different types of intervention. This is the consort diagram, just accounting for all the patients. And the final outcome was 206 patients. Sorry, those numbers don't add up. I think it was around 200, 209, 206 in the final analysis. I don't have the final analysis here to show you. Those numbers are embargoed because we've submitted them for publication. So I have to refrain from sharing the data, but I can tell you that we were quite optimistic at the results, and they've been submitted to a high impact journal, hoping for publication. And because that trial was registered with the FDA in advance, in fact, to be able to be funded to receive the NOA for that trial, not only did we have to get approval from study section and the program officer and the NIH, but we also had to receive approval from the FDA to be able to proceed with the trial. And that was from getting a positive score to running the trial, it took a full year. So again, the old Coolidge refrain, persistence pays. We really persisted with this one. But along the way, we were able to look at other patient populations. I mentioned earlier on that not only do patients with depression struggle to respond to antidepressant medications, but patients with depression who have comorbid psychiatric conditions struggle to respond to medications. And one particular comorbid or co-occurring psychiatric disorder is something known as borderline personality disorder, which is associated with significant suicidality as part of its depression and symptomatology. And so based on our findings of magnetic seizure therapy for suicidal ideation, and together with some outstanding colleagues, including Jenna Trainer, who's pictured up at the top right, we conducted a clinical trial of magnetic seizure therapy, open label, in patients with depression, co-occurring borderline personality disorder. Again, population of patients who engages in suicidality and oftentimes self-harm as well. We randomized we enrolled a relatively small group of patients that were difficult to recruit for, but our findings were striking. Not only did their suicidality decrease considerably, but the depression symptoms also decreased considerably. And this is not only in borderline personality disorder patients, but we had a comparator group. The comparator group was patients who received what's called dialectic behavioral therapy, which is the gold standard psychotherapy for these patients. So in one group, these patients weren't randomized, they self-selected to treatment, but the group that received magnetic seizure therapy plus dialectical behavioral therapy produced superior rates of suicidality and superior depressive symptom improvement compared to those who received dialectic behavioral therapy alone, who basically didn't change over the course of this trial. There was other conditions that we're also looking at. Schizophrenia is a population of patients who are also very, very resistant to not antidepressant medications, but also antipsychotic medications. And electroconvulsive therapy has been used in that patient population. This is a study by George Petridis demonstrating the clinical efficacy of electroconvulsive therapy in clozapine resistant, which is like saying treatment-resistant depression, except that in schizophrenia, we consider them resistant when they've not responded to antipsychotic medications. This was an even further level of resistance in one of our most effective antipsychotic medications, clozapine. So these are patients who did not respond to clozapine. And when combined with ECT, these patients did substantially better. We conducted a trial using magnetic seizure therapy and also found significant rates of improvement in their psychotic symptoms and almost no change in their cognitive symptoms as would be predicted. Which led to another trial. This one was funded by the Canadian Institutes of Health Research. It's ongoing. Our target population was 160 patients with schizophrenia, randomized to either bilateral electroconvulsive therapy or magnetic seizure therapy. And again, that's ongoing at the Center for Addiction Health in Toronto. And it was led by me. I handed it off to a good friend and colleague, George Fusius, who's together with Dan Bloomberger is running this trial right now. So I've already described a lot. I won't get into all these details because this is a bit much to handle, but what I will let you know is we've been managing to evaluate neurophysiology not only at the cortical level, but at the subcortical level in relation to this intriguing area of the brain called the subgenual cingulate, and showed that stimulation of the subgenual cingulate actually also helps predict therapeutic response. And we're continuing to evolve those trials in large trials that have been funded by the NIH since I moved here. So in summary, neuromodulation treatment is effective when pharmacology and psychotherapy are not. Both repetitive transcranial magnetic stimulation and magnetic seizure therapy are important treatments for depression and suicidality. It's also an important treatment. Magnetic seizure therapy specifically can also be used not just in depression, but also in schizophrenia and suicidality and borderline personality disorders. We're starting to narrow in on the mechanisms, which include inhibitory neuroplasticity in the prefrontal cortex and potentially subcortical circuitry as well. And we have many multi-center trials currently underway to disentangle those effects, specifically as it relates to the underlying neurobiology. Appreciate you all participating today. And again, thank you for the invitation. It's an honor to present to this group, and I'm happy to take any questions.
Thank you for this excellent talk, Professor. One question came in. And you got the question. What is your insight regarding the wearables in depression monitoring?
That's a good question. So wearables are only now coming to center stage. I think there's going to be a huge opportunity to look at. I just spent a lot of time talking about physiological, that is brain physiology targets, but there might be things that are easier to monitor, including steps, including heart rate variability. There's been a lot of work done on heart rate variability and its ability to predict TMS treatment response. The results are promising. I wouldn't say they're ready for prime time, but they're definitely promising. And I think there's going to be an evolution of these wearables when it comes to large scale clinical trials to help identify relatively simple markers through which we can treat depression.
Thank you. Also, what are some challenges and opportunities in this wearable space for depression?
The challenges are receiving funding. The wearables, I think, need to be incorporated in some of our large scale clinical trials, but it's a bit of a dance whether or not we incorporate them when there's not direct levels of funding for them. So we're here at UCSD, we're trying to incorporate wearable design. We did this in Toronto where we conducted clinical trials of an electronic based form of psychotherapy to predict treatment response in depression. That was relatively easy to do because we didn't have many funding restrictions and we had a lot of philanthropic funding to support that work. When it comes to NIH, sometimes they won't pay for the additional wearable feature, and there's not a lot of requests for applications for wearable devices just yet. And so I imagine that there are groups currently looking at this key question, and we plan to look at it in a little bit more depth as well.
Thank you. Are there any markers that would lead to depression early stage? For instance, if you have increased levels of cortisol or some mood swings from job loss, family loss, that would have biomarkers that you can predict so the person, doesn't go into the suicide path.
Yeah, not a lot right now. I mean, there's been a lot of promise in this area. Cortisol was looked at very, very carefully. The HPA axis was looked at very carefully back in the early 80s. And people found that this dexamethasone suppression test was seen as a real promising indicator, but in the end, it didn't really pan out and never made it to the point where we were using it for routine clinical purposes. But I think, the world is evolving. Our scientific techniques are evolving. We've had breakthroughs in relation to new peptides being developed and new treatments being developed. I would not be surprised if in the next decade or so, given the scale of innovation, given all the AI that we have now in our fingertips, that we start to identify um, uh, physiological targets of response.
Thank you. Regarding borderline personality disorder, do you think it's a chemical imbalance, or what are your thoughts on that?
Um, I think that all brain disorders are chemical imbalances. I mean that sincerely. I just think that we just don't know the chemical imbalances. We do know that borderline personality disorder, 9 times out of 10, is associated with significant amount of trauma. And that trauma results in a change, quite frankly, in neuroplasticity in the brain. And we've seen those neurophysiological changes that take place. So is it a chemical imbalance? It is a chemical imbalance. How do we target that chemical imbalance? I think that is a TBD. But there's no question that trauma affects the brain and can have profound consequences on brain physiology, as we all know, brain structure as well. And so that leads to some subtle features, including cognitive impairments, including significant psychiatric symptoms, which are not so subtle. But trying to link those features to underlying neurochemistry is, I think, an endeavor that is going to take us a few more years to uncover.
Thank you. Also, most borderline people refuse treatment or even participate in studies. How do you find and recruit these?
It's a great question. It's a great question. One of the challenges we face in psychiatry is a patient population that is not always willing to participate in our research trials. And you can imagine that if we have someone who's incredibly psychotic, someone who is suicidal, someone who is incredibly aggressive and at risk for self-harm, those patients are real hard to get into clinical trials just because of the way we structure our clinical trials. We need them relatively medically stable. And so oftentimes we don't get a truly representative sample in our trials because they need to be relatively stable. But what we see is far from that in the clinic. And so finding ways of getting severely ill patients into our clinical trials, including borderline personality, times is a major barrier to making breakthroughs, because if we don't have the science, we don't have the breakthroughs. So it's a challenge that our field really struggles with. How we're going to overcome it, boy, that's a tough question, and probably deserves several grand rounds onto itself. But I will tell you that sometimes you have to have multiple sites with multiple investigators and relatively small numbers per site. But when you aggregate those subjects across North America or globally, I think you can start to answer those key questions. So scale is everything.
Thank you. Also, what is neuroplasticity? From what I read, is the way your brain forms. You mentioned
Yeah.
several times. What is neuroplasticity?
So neuroplasticity, colloquially, is a rewiring of the brain in response to environmental adaptation changes. So neuroplasticity at a neurophysiological level involves repeated entrainment of neurons, and those neurons, when they're entrained, they fire together. When they fire together, they become hardwired together. And so it's a subtle rewiring of the physiology of the brain that leads to enhanced output. And one of the key drivers of neuroplasticity, sorry, one of the key consequences of neuroplasticity in the brain is memory. Memory is a behavioral example of what neuroplasticity does. Learning requires multiple inputs from different regions of the brain onto key termini down in the hippocampus, which then lead to enhanced recall information and output. So neuroplasticity is the physiological equivalent of memory in the brain.
Thank you. Two final questions. One is, um, what is happening at the cellular level when you apply magnetic field? And I noticed you're applying pulses, and magnetic field can be even have varying magnetic fields can be also be electric field. But what's happening at the cellular level? Do you know?
Yeah, what's happening is I think we stimulate neurons to fire, and those neurons are happening at a not at a direct pyramidal level, but at an interneuronal level. And the physiology behind that is complex, but because magnetic fields orient and cause activation in a certain way, it's the horizontally oriented neurons, which are the most superficial in the cortex, that are actually activated with the lowest intensities. So those little interneurons are more sensitive to magnetic field changes than our descending pyramidal neurons. So when we repeatedly stimulate, we stimulate those tiny little interneurons in the cortex, and we cause them to fire both excitatory and inhibitory. And it actually is the inhibitory ones that tend to be more persistent. And that's why we think inhibitory neuroplasticity may be key to uncovering the therapeutic effects of magnetic stimulation in the cortex. So ultimately, we're stimulating neurons in the cortex. Those neurons have both excitatory and inhibitory effects. The excitatory effects cause output, and the inhibitory effects cause therapeutic change, we think, in depression.
So I know about electrical stimulation where you're sending charge to the neurons, but magnetically, are you applying, is it force or is it affecting the electrons or ions inside the neurons?
It affects ions and it affects the ions transsynaptically through across the entire axon, but most consistently across the dendrite and a little bit across the cell body.
Thank you. Final question. It's fascinating that the techniques you show, the magnetic electrical stimulation, can this be extended to other areas like dementia or Alzheimer's or Parkinson's?
Great question. Great question. The answer is yes. In fact, those trials are currently underway, and we think that significant impairments in neuroplasticity underlie um, Alzheimer's and early dementia. And the reason for that is because, as I mentioned, neuroplasticity and memory are part and parcel with one another. And so if you have aberrant neuroplasticity, that significantly aberrant neuroplasticity, that could lead to dementia. So the idea of regenerating those neurons and causing reactivation of those neurons through magnetic pulses is quite appealing, and those trials are currently underway.
Thank you so much. Thank you for your time and answering all these questions, sir. I
You're
appreciate
welcome.
your
Thank you
time.
for the invitation.